recorded and it will be later posted on our website. There will be a Q&A session at the end, so please add your questions in the Q&A feature so it's easier to moderate. And now I'll let Dr. Lude introduce our speaker for today.
Thank you, Danielle. So it's my great pleasure to introduce Dr. Mariana Kaplan. Dr. Kaplan did her medical training in Mexico and subsequent fellowship at University of Michigan, where she pursued a career as independent investigator studying cell death and vascular disease in lupus. But then in 2010, she made this novel discovery of low-density granulocytes in lupus. And since then, she has become this leading expert in neutrophil biology in the lupus pathogenesis. Dr. Kaplan is currently a senior investigator and the chief of the systemic autoimmunity branch, as well as the deputy scientific director over at NIAMS. And Dr. Kaplan has authored more than 200 peer-reviewed publications, several reviews and book chapters. And she has also received several prestigious awards, not least the ACR Distinguished Basic Translational Investigator Award, as well as more recently being elected to the National Academy of Medicine. So with that said, Dr. Kaplan, I'm very grateful for you taking the time to share with us some of your work today, and much looking forward to your presentation.
Thank you very much, Christian. And again, I very much appreciate the kind invitation to be here today. I was there, I was trying to remember, Keith had invited me maybe 10 years ago. What was the last time that I had presented here? So it's great to be back. And as Christian mentioned, I'm going to focus today on some discussion of what we believe is the potential role of neutrophils in a number of systemic rheumatic diseases. I tried to make the talk a mix of basic, translational, clinical, so hopefully everyone can find something of interest, and I look forward to the discussion. So you all know that systemic autoimmune diseases are complex and very heterogeneous. However, we know now that they do have some commonalities in that they represent multi-step processes triggered or facilitated by a combination of genetic and environmental factors, many of these poorly understood. And what we see is that a variety of environmental stochastic events throughout an individual's lifespan will lead from this susceptibility phase to a subclinical phase or preclinical phase, where in many of these diseases, we have the development of profound, aberrant, innate, and adaptive immune responses, where for some of these diseases, pretty much all cell types from the immune system you look at are abnormal. And again, through a variety of subsequent stimuli and genetic interplay with environmental triggers, at some point, individuals predisposed to develop systemic autoimmunity will develop autoantibodies that target both cellular and extracellular autoantigens. And as you know, for example, in the case of lupus and rheumatoid arthritis, we do know that these autoantibodies can be present for many years before clinical diagnosis is made. But at some point, again, after a second, third, or fourth hit, there will be the development of the acute clinical manifestations characteristic of the specific systemic autoimmune disease. And many of these individuals that are diagnosed with systemic autoimmunity go through periods of remission and flare. And as our patients live longer and we have managed to control their disease better, we are now encountering more and more the chronic complications that in many cases are the result of prolonged exposure to chronic oxidative stress, including cardiovascular disease, bone damage, fibrosis, and cancer. Now, a big question for us studying autoimmunity over the last several decades has been, how do these intracellular autoantigens become immunogens that can trigger and maintain strong, prolonged autoimmune responses? And the dogma for many years was that dysregulation in apoptosis, being more enhanced apoptosis of immune cells and other cells in individuals at risk, coupled to impaired clearance of apoptotic material, was a significant event that in those predisposed individuals would fuel the fire to induce both innate and adaptive immune dysregulation. And that's probably the case in some patients, but we now think that this is an oversimplified view. And this is understandably so because until much more recently, we knew far less about other types of cell death. But as you know, we now know that cells can die in many different ways. And it's not only whether the cell will die or not, but how it dies, where it dies, what's the cell that is dying, and what is the surrounding microenvironment of the dying cell that may be key in driving a tolerogenic versus immunogenic response to the dead cells. And in that regard, my group has been particularly interested over the last decade or so in understanding how neutrophils live and die and how that may be an important phenomenon in the context of systemic autoimmunity. And just to pause here for a second, I would like to emphasize that the field of neutrophil biology, which really lacked, our understanding of neutrophil biology really lacked compared to what we know about other cell types, both innate and adaptive immune cells, in part because of technical issues, and also because these cells are very hard to study because they live a very short period of time. They're very sensitive to stimuli. They die very easily, etc. They're terminally differentiated. They don't divide. So it's very hard to study them. But through recent technological advances, I think there's been a significant shift in how we perceive neutrophils. And it has shifted from thinking of these cells as this monolithic, boring cell type that is there to sense microbes, to be a very important role as a first line of responders against microbes. They go to a site of injury, they kill the microbe, and then they die, to placing them playing a much more central role in shaping the immunologic landscape in both health and disease. So I won't have time to go through all of this, but just to emphasize that in addition to their important antimicrobial role, we now know that neutrophils play many other important roles, both under conditions of health and disease. And these may involve, for example, their important role in wound healing and normal coagulation. They do have some poorly understood immunosuppressive functions, on immunoregulatory functions. They play important roles in the resolution of inflammation. They can interact with pretty much all other immune cells and can play important roles in shaping how these other cells will behave. And in the context of autoimmunity, there are certain aspects of neutrophil biology that we have been particularly interested in studying. One, which I will discuss during my talk, has to do with this concept of plasticity and cellular heterogeneity, which is really a much more recent finding for neutrophils than for other cells. We used to think all neutrophils are the same, but now we know that there is this concept of cellular heterogeneity within neutrophils and what this may mean in the context of autoimmunity. And the other concept that I will discuss is how specific types of neutrophil cell death may be particularly conducive to the development of immunogenicity and organ damage in the context of autoimmunity. And I will talk particularly about lupus, but I will also spend some time on other diseases. And then what I find fascinating is that we're learning more and more how neutrophils are regulated. And for example, we now know that neutrophils actually have their internal circadian rhythm. And they can also be modulated by external factors that modulate circadian rhythm. But this goes to tell us that a neutrophil in the morning will look much different than a neutrophil at night. And when we study them may be very relevant, but also this may be linked to the development of certain complications. For example, morning stiffness in rheumatoid arthritis, or why heart attacks are more common in the morning. There are some links with these differences in circadian rhythm in neutrophils and how their function may vary from day to night. There is a lot of interest now in how the microbiome may be modulating neutrophil biology. I will talk a little bit about our work on how sex hormones may regulate how neutrophils behave. Certainly, neutrophils in neonates are much different than neutrophils in the elderly, and how the life cycle modulates neutrophil phenotype and function. It's an area of interest that remains to be better characterized. The role of genetics in modulating how neutrophils behave, and very interesting how the tissue microenvironment may change how neutrophils behave. And there was a very interesting paper from Keith Elkon's group on this in the context of lupus and how neutrophils priming the skin may have a role, for example, in developing kidney damage in the context of lupus. And so it's already been almost 20 years that Arturo Sichlinski at the Max Planck Institute of Infection Biology in Berlin described this new type of antimicrobial strategy driven by the neutrophils, whereby when a neutrophil encounters certain danger signals, they undergo a very dramatic process where in a very schematic, simplistic way, through still not fully understood downstream signaling cascades, the membranes of the granules and the membrane of the nucleus of the neutrophil will dissolve. And then this leads to the neutrophil, the cytoplasmic granule and nuclear content mixing in the cytoplasm. And there is activation of proteases and of peptidyl arginine deaminases, or PADs, that, as you know, are important in inducing citrullination of proteins, including histones. And when this happens, the chromatin of the neutrophil profoundly decondenses. And you can see that at some point, the nuclear material occupies the whole diameter of the cell, mixed with these granule proteins. And this leads at some point to the disruption of the cell membrane of the neutrophil and the extrusion of this meshwork of nuclear material bound to granule protein material that was termed neutrophil extracellular traps or nets, and the mechanism of cell death at that point was termed netosis. And while initially described in the context of infections, we now know that a variety of sterile inflammatory stimuli that are highly prevalent in systemic autoimmune diseases also have the ability to induce net formation. And this includes cytokines, complement components, autoantibodies and immune complexes, but also crystals, urate crystals, cholesterol crystals, certain drugs that are known to trigger autoimmunity, and platelets. They all have the ability to induce nets under certain conditions. And what is very important to mention is that nets carry a lot of concentrated autoantigens that are known to be key targets of the immune system in systemic autoimmunity. So for example, there's a lot of double-stranded DNA and histones in the nets, but also myeloperoxidase and PR3, key targets in ANCA vasculitis, are highly enriched in nets. And then because PADs are activated during net formation, there is the extrusion of citrullinated autoantigens that are known to be key targets of the immune system in rheumatoid arthritis. And what we also know is that this process appears to be operational in vivo, because when we have looked at tissues from patients with a variety of systemic autoimmune diseases, we find evidence of netting neutrophils in these tissues. Skin, kidney, placenta in the case of lupus, the synovium in the case of rheumatoid arthritis, different organs in inflammatory myopathies, for example, and certainly in ANCA vasculitis. And there is now the realization that in many of these diseases, we do see an imbalance between net formation and net degradation. And so that the idea here is that this net material can be present for longer time in people with autoimmunity, and this may contribute again to fueling the fire and inducing pro-inflammatory responses in individuals at risk. So and there's also a chicken and egg question, right? We do have evidence that net formation carries various autoantigens that may be targeted by autoantibodies or may promote the formation of autoantibodies. We also do know that a number of autoantibodies and immune complexes, a characteristic of certain autoimmune diseases, have the ability to induce nets in healthy neutrophils. And this was work we did with Christian Luth and Keith Elkon years ago, where we found that lupus RMP immune complexes have a particular ability to induce net formation. We have also found that, for example, anti-citrullinated by-19 antibodies purified from patients with rheumatoid arthritis also have the ability to induce nets, as do ACPAs. Others have found that ANCAs purified from ANCA vasculitis patients similarly can induce net formation. And we found a few years ago, in collaboration with Ingrid Lundberg, Andy Mamin, and others, that anti-MDA5 antibodies from patients with inflammatory myopathies, but not, for example, other myositis-specific antibodies have the ability to induce NETs by healthy control neutrophils. Now, why some antibodies and not others have this ability is something that remains to be better understood and characterized. So the field of NETs and their role in health and disease has really exploded over the last several years, and there is now, I think, fairly convincing evidence, at least in the context of in vitro studies and animal studies, that NETs may be implicated in the pathogenesis of a variety of autoimmune and autoinflammatory syndromes. And here I'm listing, and this is a non-exhaustive list that includes, the usual suspects, as I mentioned, of systemic autoimmune diseases, but they've also been implicated in the pathogenesis of gout and in a variety of autoinflammatory syndromes, including DADA2, the recently described VEXA syndrome, PAPA, and in hidradenitis suppurativa, we have found evidence for that. And beyond autoimmunity and autoinflammation, there is also evidence that NETs may be implicated in the pathogenesis of a variety of acute and chronic inflammatory conditions, including atherosclerosis, cancer, diabetes and metabolic syndrome. And more recently, as you know, there's been a lot of interest in the putative role of NETs in some of the complications of COVID-19. And schematically, what we have proposed is that neutrophils, and especially in our hands, certain types of neutrophils, when they form NETs, these NETs may have immunomodulatory and vasculopathic capabilities that may drive complications in autoimmune diseases. So for example, we have found that NETs have the ability to stimulate the NLRP3 inflammasome machinery in macrophages, which may lead to enhanced production of pro-inflammatory cytokines that in the context of kidney damage in lupus and other complications may be quite relevant. There is evidence from various groups that NETs are strong inducers of type 1 interferons by plasmacytoid dendritic cells, which may be quite relevant in lupus and other type 1 interferon-driven diseases. We do know that NETs have the ability to activate both T cells and B cells, and this may be relevant again in several autoimmune diseases. And as I mentioned, we've been quite interested in understanding the role of NETs in inducing vasculopathy and immunothrombosis in the context of autoimmune diseases. And we do know that NETs, in particular, NETs induced by certain types of neutrophils I will talk more about, are very good at damaging the endothelium by inducing endothelial cell death. But NETs are also very important both in homeostasis and disease in driving thrombosis. They can activate platelets, they can activate coagulation factors, and actually some of the proteases present in the NETs can inactivate certain anticoagulants, therefore shifting things to a more procoagulant state. The other interesting aspect of NETs we've been studying is that NETs may serve as messengers that carry signals to other cells. And there's been evidence by several groups, including ours, that, for example, NETs can carry histones present in the NETs, and especially modified histones may have the ability to damage muscle cells, both vascular smooth muscle cells in the context of vascular disease. But as I'll mention later, we also found evidence that NETs can also damage myotubes in the context of voluntary muscle, and this may have a role in the context of inflammatory myopathies. We do know that NETs also carry a variety of small RNAs, and we recently found that some of these small RNAs have the ability to be taken up by endothelial cells, and when this happens, these endothelial cells undergo pro-inflammatory changes that may be conducive to vasculopathy. And as I'll mention later, we do have also some evidence that in the context of rheumatoid arthritis, modified proteins present in these NETs have the ability to change the phenotype and function of synovial fibroblasts and induce pathogenic adaptive immunity. So this is an ongoing area of research, and we need to understand much better how these NET components get internalized and how that changes the phenotype of target cells. But it's an additional mechanism that by which neutrophils may promote changes in other cells. Now, what about genetic polymorphisms and how may this be linked to the development of abnormal NET formation, and how do we link this to lupus. So it's interesting that many of the polymorphisms that have been associated with enhanced risk to develop lupus have also been linked in recent years to a higher ability to form nets. And one may think, okay, well, if you're studying this in the context of a lupus patient, what comes first, right? Is this genetic polymorphism really leading to more nets, really, and that in turn playing a role in inducing SLE? And that may be hard to dissect, but for example, in the context of IRF5, which is a gene that has been linked to enhanced risk for SLE development, Betsy Barnes' group has shown that even in individuals who have not developed lupus, but that have the risk allele, the IR5 risk allele associated to lupus development, those healthy people with this polymorphism have a higher ability to form nets. Suggesting that, at least in some cases, the genetic polymorphisms may promote risk in part because they may lead to dysregulation in neutrophil biology. And we recently described that in the context of STAT4 as well, which is doing many other things. But one thing that we have found, for example, is that people with the STAT4 risk allele for lupus have an enhanced ability to form nets. The other factor that may be important in in determining why certain people form more nets than others is whether an individual is a man or a woman. And years ago, we partnered with David Furman and Mark Davis' group at Stanford, looking at if you take healthy young adults and you look at their peripheral blood transcriptome, what's different between young females, adult females, and young adult males? And the most striking differences were in genes that are associated with neutrophil biology. And long story short, what we found is that males overall have a more immature neutrophil phenotype than females. So this was an enhanced immature neutrophil signature in the males that translated to finding that female neutrophils are overall more mature and more activated. And this was associated with finding that in healthy young adults, females tend to form more nets than men, either spontaneously or upon exposure to microbial or sterile inflammatory stimuli. So this may suggest that one of the potential mechanisms by which females, and of course it's not the only one, but by which females are more likely to develop autoimmunity may be through higher ability to generate autoantigens through net formation and higher neutrophil activation. And so we then went on and expanded on these studies where we purified neutrophils from peripheral blood of both young adults, females and males, and look at their gene transcriptome. And one of the most striking differences between females and males was that females tend to have higher levels of type 1 interferon regulated genes than young males. And importantly, this was replicated in more than one cohort. This, as you can see, is an interferon score of the female neutrophil versus the male. So overall, they have a significantly higher interferon score. But also, importantly, this appears to be neutrophil specific. When we did single cell RNA sequencing and look at other cell types, these differences between females and males in neutrophil type 1 interferon regulated genes was not seen in other cell types, suggesting that there's something unique to the neutrophil signaling mechanisms that may be different between males and females. And we have done a lot of work trying to define what this may be. It doesn't appear to be because females have higher levels of circulated type 1 interferons or higher levels of the type 1 interferon receptor in their neutrophils. It is more that the signaling responses appears to be higher in the female neutrophils than in the male neutrophils. And we think that this overall goes again to the concept of female neutrophils being more mature. And therefore, the signaling cascade appears to be more active in these neutrophils than in their male counterparts. And then the question became, is this hormonally driven or is this related to the X chromosome? And this is very hard to study in vitro in human samples. But what we did in an indirect way is we took neutrophils from patients with Klinefelter syndrome and tried to see, okay, if this is related to having an extra X chromosome, we would think that these neutrophils behave more like the female neutrophils, not like the male neutrophils that don't have Klinefelter. But as you can see here, the Klinefelter male neutrophils, regarding their type 1 interferon response, behave much more like the male without Klinefelter, suggesting that this is more driven by a sex hormone response than by an X chromosome response. And to confirm that, we have found that if you take prepubescent children and compare kids, boys and girls, neutrophils, and in their type 1 interferon responses, we see no difference. So again, this tends to support that this may be sex hormone driven. And we have found, for example, that metabolic responses of neutrophils differ between men and women, and this can be modulated if we add sex hormones in vitro. So this is ongoing, where we want to also recapitulate some of these findings in mice, which is not easy, but it would appear to suggest that sex hormones have a role in modulating neutrophil biology. And again, this could be an important factor in explaining why women are more likely to develop autoimmunity and yet can fight infections better. This may have relevance even to COVID-19 pathogenesis. So and now we'll try to focus more specifically on neutrophil heterogeneity and autoimmunity. And when we did these studies in healthy individuals and did single cell RNA sequencing, I think what is important to highlight is that even in healthy people, not all neutrophils are the same. And at least at the transcriptional level, we were able to identify at least 4 subsets of neutrophils that are in circulation in healthy people. And what these neutrophil subsets do is something that we're heavily investigated, but we still don't really know. But what is interesting is that among those subsets, there is only one of them that is the one that appears to really upregulate type 1 interferon regulated genes. And it is not that women have more of this subset, it is that within this subset, they tend to upregulate the interferon-regulated genes more than in males. But again, just to emphasize that not all neutrophils are the same, even in healthy people, and what these different subsets mean remains to be determined. Now, years ago, when we were trying to purify different cell subsets from lupus patients, we ran into this issue that when we were doing a FICOL gradient to get peripheral blood mononuclear cells, we were finding at that time what we call a contaminating subset of neutrophils that was pretty much present every time we were trying to purify PBMCs. And typically when you do a FICOL gradient, because neutrophils are heavier than other immune cells, they tend to go to the bottom, more closer to the red blood cells. But here we were finding a lot in the PBMC interface. And long story short, we spent a lot of time trying to figure out if these cells were just innocent bystanders, artifacts, etc. And what we concluded after many experiments was that these cells appear to represent a distinct pro-inflammatory subset of neutrophils that are present at high levels in patients with lupus. And what role they play, or they appear to play a variety of different pro-inflammatory roles. And I just want to emphasize, for example, that they tend to make more pro-inflammatory cytokines and synthesize higher levels of type 1 interferons than other types of neutrophils. At the level of the lupus bone marrow, they may be playing certain roles in driving abnormal lymphocyte ontogeny. And one of the most striking features of these cells is that they come out of circulation exquisitely primed to form nets. So typically when you get a neutrophil out of the blood, you need to stimulate it with something pretty strong to make it form nets in vitro. But in the case of the lupus low density granulocytes, there is nothing that we can really add to make them form more nets. They have maxed out their ability to form these structures. And one of the features that we think are very key in what's the consequence of these low density granulocytes or LDGs making more nets is that these nets appear to have a very high capacity to damage the endothelium and promote vasculopathy. And from lupus, we have been able to see that in other systemic autoimmune diseases and some autoinflammatory syndromes, there is also the presence of these low density granulocytes. But these cells are not just present in any inflammatory condition. We haven't found them in some of the ones we have looked at, but we have found them, for example, in ANCA vasculitis, in Lipamisol-Induced Vasculitis, in psoriasis, in idiopathic inflammatory myopathies. Jason Knight found them in antiphospholipid syndrome, and we also have described them in PAPA syndrome and in DADA2 syndrome. And something that these conditions may share as a commonality is that they are all associated with significant vascular damage or vasculopathy. And we think that this presence of low-density granulocytes may be potentially a unifying factor in the promotion of vascular damage in these diseases. So what are these low-density granulocytes, and can we tell that they are really different from other neutrophils that may be present in lupus or in healthy people? So this is just to show you that if we do bulk RNA sequencing on the left, and we purify low-density granulocytes from lupus patients in red, normal dense neutrophils from the same lupus patients in blue, and healthy control normal dense neutrophils, and compare the transcriptome, neutrophils from lupus patients that are normal density are really not that different from healthy control neutrophils at the level of the transcriptome. But in contrast, the low-density granulocytes appear to have a very different gene signature. So this suggests that at least transcriptionally, they behave differently. When we have done single-cell RNA sequencing of the lupus PBMCs, we have seen that these low-density granulocytes appear to be the main cells responsible for the enhanced type 1 interferon gene signature in lupus patients. Not that other cells aren't important, but as you can see here, this appears to be driven primarily by the low-density granulocyte fraction in PBMCs. Now, what does this mean in the context of neutrophil type 1 interferon interactions remains to be determined, but we do think this is an important type of crosstalk in the context of autoimmunity. The other interesting observation we have made, and this is a very busy slide, please don't look at all the details, but just to highlight that we then move to the proteome of these cells and try to see, okay, does the protein cargo of low-density granulocytes vary or is it different from healthy control neutrophils or from neutrophils that are normal density from lupus patients? And can this give us any clues about why they behave in certain ways and about their function? So what was striking is that when you look at the proteome of lupus LDGs, it is different in several aspects when compared to the proteins that are present in other types of neutrophils. And one of the most striking differences was in the expression of proteins that regulate the biomechanical properties of the cell, their ability to perhaps adapt and change their shape when they are traveling in blood vessels or where they are entering tissues. And something we found doing a technique called RT-DC, which is a type of flow cytometry assessment that allows you to see how stiff a cell is, how it may adapt as it travels through blood vessels, is that the low-density granulocytes have impaired biomechanical properties when compared to other types of neutrophils. And to see whether that has some functional implications, or to start to see if these may have some functional implications, we designed this 3D mimetic where you put a neutrophil here and then you quantify how long will it take for the cell to travel, kind of mimicking what small capillaries may look like, and then exit the mimetic. And what we found is that compared to healthy control neutrophils or lupus neutrophils of normal density, the low-density granulocytes tend to have a much higher retention in blood vessels than the other cell types or the other neutrophil subsets, suggesting that these cells, because they have different biomechanical properties, may tend to get trapped in small blood vessels. And this could have important consequences in the context of vasculopathy or organ damage or hypoxia development in small vessels when compared to what other neutrophils may do. And this was not because they may tend to stick more to endothelial cells. We did not find much difference in how these cells stick to the endothelium, but they just appear to have lower ability to travel through something that mimics small capillaries. And this is ongoing work where we want to further determine what's happening to these cells in circulation and in tissues. Now, the other question we have had is, are all nets created equal? So we probably all make nets at some point in our lives in response to a variety of danger signals. But is it the same to make nets if you have lupus or if you are exposed to just some sort of microbe? And what may be driving that? And so again, this was work done in collaboration with Christian and Keith years ago, where nets were purified from low density granulocytes from lupus patients or from healthy control neutrophils that have been induced to form nets by some sort of microbial stimuli. And when we look at the ability of these nets to induce type 1 interferon responses in target cells, while nets in general do a pretty good job at doing that, it was evident that the nets that are formed by lupus low-density granulocytes are more interferogenic, and this may have relevance in the context of diseases that are type 1 interferon dependent. And we have also found that if you purify nets from low-density granulocytes or from other sources, and look at the ability of these nets to damage the vasculature, the lupus low-density granulocyte nets impair much more the function of the endothelium that nets generated, for example, in the context of infections by healthy control neutrophils. So all of this suggested that the protein content and perhaps post-translational modifications present in the nets may be important in making a net be more immunogenic or more vasculopathic than other nets, and so that not all nets are created equal. But furthermore, and again, this was work we did with Christian and Keith years ago, it's not only the proteins, but what we believe may be quite relevant is the quality of the nucleic acids that are present in the nets and how that may induce pro-inflammatory responses. So what we found at that point, as I mentioned, is that when healthy control neutrophils are exposed to lupus relevant stimuli such as RMP immune complexes, but not to other stimuli that may not be lupus relevant, the mitochondria present in these neutrophils produce enhanced levels of mitochondrial reactive oxygen species. And because mitochondrial DNA lies in close proximity to the sources of reactive oxygen species, it becomes oxidized. And nets are then extruded carrying enhanced levels of these oxidized nucleic acids. And this appears to be a phenomenon that triggers enhanced synthesis of type 1 interferons and other inflammatory stimuli. And in fact, we found that low-density granulocytes are heavily dependent on the production of mitochondrial ROS to form nets, because when we use a mitochondrial ROS scavenger, the ability of these low-density granulocytes to form nets was significantly impaired. And this has relevance in vivo, because if we give mitochondrial ROS scavengers to lupus-prone mice, they appear to have less inflammatory phenotype, and the lupus features are decreased, including the production of type 1 interferons. So overall, through work by various groups, including the collaboration I mentioned with Keith and Christian, but this has been reported by several groups now, the idea here is that diamide net formation may be highly conducive to nucleic acid oxidation. And this is not only mitochondrial, but it is also genomic, because as I mentioned, during net formation, there is profound decondensation of the chromatin. So the genomic DNA is also less protected from oxidation. And these oxidized nucleic acids may be taken up by target cells. And there is evidence that when DNA becomes oxidized, it is less likely to be degraded by nucleases, and then more likely to be sensed by intracellular sensors of nucleic acids, such as the CGAS-STING pathway, which in turn may lead to enhanced production of type 1 interferons. So this may be one of the factors that may make this type of cell death more immunogenic than dying by other mechanisms. And this has been also supported by recent work done by Moshe Arditi's group at Cedars-Sinai, with whom we had the opportunity to collaborate. And in this case, the idea was to induce lupus using the pristine mouse model of lupus in wild-type mice or in mice that lack OGG1, which is an enzyme that repairs DNA and excises oxidized nucleotides. And so when the mice are not able to repair oxidized nucleic acids and carry a higher amount of oxidized nucleic acids, when the lupus is induced by pristine, these mice develop a more severe phenotype. They get skin disease, they get alopecia, they get inflammation in the skin, but also systemically, they get a higher type 1 interferon response. They get more levels of autoantibodies. So again, these results suggest that having oxidized nucleic acids that you cannot repair, and this perhaps triggered in part by dysregulation in net formation, may be a factor that is conducive to autoimmunity. So what about other autoimmune diseases, and can we translate some of these findings into what these cells may do in other autoimmune diseases? So when I moved to NIH, I started talking to Peter Grayson, who heads the vasculitis translational research group at NIAMS, and he had the RNA sequencing data from the RAVE trial that, as you know, was a landmark study that showed the efficacy of rituximab compared to cyclophosphamide for ANCA vasculitis. And what Peter had noticed is that although patients responded quite well to rituximab, when they were looking at the non-responders, or those who responded less, the gene signature that was associated with poor response was a neutrophil signature that pretty much overlapped completely with what we have found as the low density granulocyte gene signature in lupus patients. And long story short, what we found is that similar to lupus, in vasculitis, there are these low density granulocytes that, as in lupus, have a higher ability to spontaneously form nets. And these nets externalize a lot of myeloperoxidase and PR3. And so now we're looking at this carefully longitudinally to see whether the presence of low density granulocytes can be a useful biomarker to predict certain complications or of flares. And we are not there yet in trying to figure this out, but it's interesting that there are some similarities between the lupus and the ANCA vasculitis low density granulocytes. And interestingly, we then went and looked at specific types of drug-induced vasculitis, and that I'm talking here about, as you know, Lipamisol-Induced Vasculitis. And you all know that one of the main sources of levamisol intake is contamination of cocaine for profit, and that most of the cocaine used in the US is tainted with levamisol, and that this is a very distinct type of vasculitis, that not only because of the localization of these lesions, but also because you have a variety of autoantibodies that are part lupus, part ANCA vasculitis antibodies, antiphospholipid, etc. So it's a mixed bag of autoantibodies. And what we found years ago was that in Lipamisol-induced vasculitis, there was evidence of low-density granulocytes and enhanced net formation. And what we found is that levamisol can directly induce net formation, and it does this in part through engaging certain muscarinic receptors in the neutrophil. So that if you use a muscarinic agonist, you will be able to induce net formation in neutrophils. And actually, if you inhibit muscarinic engagement, receptor engagement, you can inhibit net formation. And the quality of the nets induced by levamisol was also different in that it appeared to be more vasculopathic than other types of nets generated through other stimuli. So this goes again to this concept perhaps of neuroimmunomodulation, and that certain drugs or even endogenous factors that we don't necessarily link to immunity may have a potentially important role in driving inflammation. And it may lead us to think more about other drugs associated to immunity that may do so in part through the induction of neutrophil dysregulation. The other disease where we had, we did some studies in the context of neutrophil dysregulation is idiopathic inflammatory myopathies. And I know Christian has also published some very nice work in the context of calcinosis and the development and in idiopathic inflammatory myopathies associated with NETs. In our case, we focus both on pediatric dermatomyositis and also in adult, and we did this in collaboration with Lisa Ryder and Andy Mamen. And what was interesting is that, as I mentioned, we found that patients with idiopathic inflammatory myopathies, both kids and adults, especially in dermatomyositis, have enhanced levels of low-density granulocytes that really are comparable to what we have seen in lupus patients. And that similar to lupus, these low-density granulocytes have a higher ability to form nets when compared to other types of neutrophils in idiopathic inflammatory myopathies. And we found that there is an association with certain autoantibodies and the higher ability to form nets or higher levels of nets in circulation. And this was particularly the case of MDA5 autoantibodies that we found correlated with the presence of nets and LDGs. And in fact, when we went in vitro and looked at different myositis autoantibodies, we found that it was actually the MDA5 autoantibodies that had the ability to induce nets in healthy control neutrophils. Whereas, for example, Jo1 antibodies didn't have that same ability. And then we went and tried to see, okay, is this operational in vivo again? And we were able to have access to different tissues from patients with idiopathic inflammatory myopathies, not only muscle, but also lungs, for example, and found evidence of netting neutrophils in the lungs and in the muscle of patients with idiopathic inflammatory myopathies. We also found an enhanced neutrophil signature in the skin of dermatomyositis patients, but not so much netting neutrophils. And we did that in collaboration with Michelle Kallenberg. And then what is the role of these nets, right? So something that we found when we did studies with Andy Mammen's group is that if you take myotubules and expose these myotubules, which are very important cells in the muscle, to nets that have been purified from patients with dermatomyositis, we found that these nets had the ability to damage the myotubules. And these appear to be dependent on the presence of citrullinated histones in these nets, because if we added citrullinated histone directly to the myotubules, we were able to induce the same phenotype. But also if we blocked the action of histones by either by antibodies, we were able to mitigate the degree of muscle damage. So this suggested to us that neutrophils and nets may play a pathogenic role in idiopathic inflammatory myopathies, and further work is needed to assess their interplay with other cells and how may we inhibit the damage, and what damage may they be causing in other organs affected in this disease. And then finally, I want to mention something about nets in the context of rheumatoid arthritis, and I will do this in a very schematic way. But long story short, we found years ago that in the synovium and in the periphery, in rheumatoid arthritis, neutrophils have a higher ability to form nets. And these nets are enriched in citrullinated autoantigens that may be recognized by autoantibodies or promote the formation of antibodies themselves. We also found that certain RA-related autoantibodies, such as ACPAs and rheumatoid factor, have the ability to induce nets. And then we found initially that if we culture nets or with synovial fibroblasts from RA patients, they induce these nets induce a pro-inflammatory response in the synovial fibroblast, suggesting that they were doing something in the joint that was activated pro-inflammatory responses. In collaboration with Christian de Moruel and my colleagues group at the University of Colorado, what we have seen is that it is interesting that even if you look at individuals at risk to develop rheumatoid arthritis or in relatives of people that have rheumatoid arthritis and that they may be themselves at risk, there is evidence of an association of nets being present in the sputum of these patients at risk for RA in association with the development of both IgA and IgG ACPA responses. So the hypothesis that RA may initiate in the lung may be linked in part to some enhancement in net formation at the level of the airways, and whether that plays a role in exposing autoantigens and driving ACPA responses remains to be fully demonstrated, but at least there is some correlation there that appears to be interesting. And then the other factor that I want to emphasize is that we believe these neutrophil synovial fibroblast interactions in the RA synovium may be conducive to pathogenic adaptive immunity. And what we found a few years ago was that components of the nets can be internalized by synovial fibroblasts. And that leads to the upregulation of MHC class II in the synovial fibroblast, and therefore endows these stromal cells with antigen-presenting cell capabilities. And indeed, we found that these synovial fibroblasts can present citrullinated antigens that are present in the nets to the adaptive immune system, activate T cells, and in vivo promote ACPA responses. So this would suggest that there is this crosstalk between neutrophil nets, stromal cells, synovial fibroblasts, and the adaptive immune system that may have some pathogenic role in the context of rheumatoid arthritis. And finally, I would like to bring it a bit more into the clinic and say, focusing more on the vascular damage part of the story. And as you know, lupus is associated with profound vascular damage. And at the NIH Clinical Center, we established this cardiovascular lupus cohort where we follow patients prospectively and assess for subclinical development of vascular disease in association with potential biomarkers. And in many of these patients, we do FDG PET-CT to look for arterial wall inflammation. And we have found that many lupus patients have enhanced vascular inflammation. And this associates with a higher incidence of non-calcified coronary plaque burden. So young lupus patients have a lot more of or maybe unstable plaque, non-calcified plaque in the coronaries than young healthy females. So when we have used these as surrogates of vascular damage, we have been very interested in trying to establish what is the pathophysiological alliance between components of the innate immune system that may promote vascular disease. And what we think at this point is that lupus represents some sort of extreme phenotype where both enhanced type 1 interferon responses and dysregulated neutrophil function and net formation may converge at the level of the plaque to create this profoundly inflamed environment that may exacerbate inflammation, plaque formation, thrombosis, etc. And what hints do we have that this is operational in vivo in humans? Using the lupus cohort I mentioned, we have been looking at what factors associate with both enhanced vascular inflammation by FDG PET-CT, dysregulation of endothelial function, and enhanced coronary plaque. And what we have seen is that the levels of low-density granulocytes in lupus patients significantly associate with having more non-calcified plaque burden. And this persisted after adjusting for multiple confounders, after adjusting for Framingham risk factors. If you have higher levels of LDGs, you are more likely to have more plaque formation. And if we look at the low-density granulocyte gene expression, we have seen that those that have an LDG signature are more likely to have higher levels of non-calcified plaque and or higher levels of vascular inflammation. So although this is not proof of causality, it does suggest an association between these dysregulated neutrophil responses and vascular damage in lupus. The other thing we have found is that this enhanced net formation may be pathogenic to the vasculature by oxidizing lipoproteins. So long story short, what we found is that some of the enzymes that are present in the nets from LDGs have the ability to oxidize the good cholesterol, HDL, in a region-specific manner in a way that alters the function of the HDL. So this HDL goes from being anti-atherogenic to being pro-atherogenic and pro-inflammatory. And this appears to be driven in part by these nets. And so how do we target nets in a way also to show whether our hypothesis is correct or not? So there are a variety of different ways by which we could envision targeting nets. Some are more specific than others, and ideally we want to find strategies that won't completely alter the antimicrobial ability of the neutrophil and predispose individuals to bad infections and enhanced mortality. So one mechanism what we're still exploring is whether by inhibiting paths, which as I mentioned are implicated in net formation, we may be able to mitigate lupus phenotype or vascular damage. And all these studies have been performed in animal models, and just summarizing quickly, we have found that both through genetic manipulation or through chemical inhibitors of paths, we have seen in various animal models that we can inhibit lupus phenotype, immune dysregulation, and even atherosclerosis in certain models. Now, not all models of lupus have resulted in improvements when you inhibit paths, which goes against to pinpoint or to highlight that this is a very complex syndrome and that whether the model is more driven by innate immune dysregulation versus adaptive may be key. But there are a number of companies that are exploring the role of PAD inhibitors, at least in the preclinical level, and we'll have to see if these compounds are deemed to be safe and effective, whether these will move into the clinic at some point. I would like to mention a couple of studies we've recently published, and then I'll conclude, trying to test the hypothesis that modulating net formation of the crosstalk between interferons and nets may have a therapeutic role. So this was done in collaboration with AstraZeneca, and the idea here was, and using anifrolumab, which, as you know, was recently approved by the FDA for certain types of lupus. And the idea here was, okay, if type 1 interferons may be priming neutrophils for NETs, and NETs, we think, and type 1 interferons may be inducing vasculopathy, does the inhibition of type 1 interferon signaling have any role in reducing readouts of vascular damage? Now, this was tricky to do because by the time we established a collaboration with AstraZeneca, the study was ongoing. It was actually concluded at the time. So we didn't have access to fresh cells. We couldn't do vascular function tests in these patients, but we did have access to serum and plasma from both at baseline and also after 1 year of treatment. And we decided to compare the placebo arm versus the 300 milligram anifrolumab arm. And what we asked, because we only had serum and plasma, was, can inhibition of type 1 interferon signaling mitigate net formation, which we could measure in the periphery, or alter this ability of nets to oxidize lipoproteins and change the function of the HDL, which from work from Barbara Han and Maureen McMahon and others has shown that can be a risk factor for vascular disease in lupus. And so what we found, and here is just to show you that we can find net remnants in circulation of lupus patients being elevated. When patients receive anifrolumab, we found that there were significant decreases in circulating nets in the anifrolumab group, while in the placebo group, if anything, the net levels had gone up after comparing them at 1 year. So suggesting that blocking type 1 interferon responses, independent of disease activity, can lead to decreases in net formation. And this also translated into finding that if you look at HDL function as measured by cholesterol efflux capacity, HDL function in the anifrolumab group significantly improved. And this may not look as a lot, it's about 17%, but this is a big improvement when you see what other drugs such as statins or other cholesterol modulating, lipoprotein modulating drugs can do, while there was no significant improvement in the placebo arm. Suggesting that blocking type 1 interferon responses, perhaps through modulation of net formation, can lead to altering lipoprotein function, which may have beneficial consequences in vascular risk in lupus. And finally, the other approach we recently followed was what happens if we inhibit JAK-STAT signaling pathway. So as you know, this is a fundamental pathway by which many cytokines signal. And type 1 interferons use the JAK-STAT pathway to signal downstream and exert their effects on target cells. But also neutrophils have a very active JAK-STAT pathway, and many cytokines crucial for neutrophil development and function signal through this pathway. So we asked whether by blocking JAK-STAT pathway, we were going to be able to see changes in neutrophil dysregulation in patients with lupus. And previously in animal studies we did in collaboration with John O'Shea and Massimo Gadina, we had found that tofacitinib, one of the JAK inhibitors, can improve lupus phenotype in mice, but also interestingly improve vascular phenotype and decrease net formation in these mice, both in vitro and in vivo. So we recently published the results of a study where we gave tofacitinib or placebo to a small group of lupus patients. This was really a very exploratory safety study. So we gave it for about 2 months, and then we followed the patients for another month. And the drug was safe, at least during this short period of time. But I just want to highlight a couple of things. One was that during this short duration of treatment, low-density granulocytes significantly decreased in the tofacitinib-treated group, but increased in the placebo-treated group. And this persisted even after we stopped the drug. And also that levels of net formation also significantly decreased while patients were on the tofacitinib group, but not in the placebo group, suggesting that this drug may have the ability to modulate neutrophil function in lupus patients. And not surprisingly, we also saw decreases in the type 1 interferon signature while the patients were on tofacitinib, but also over a very short period of time, which was quite striking, HDL function significantly increased in the lupus patients on tofacitinib, but not in the placebo group. And this translated to significant changes in vascular health that were arterial stiffness, as measured by this CABI test, increased by 12% while the patients were on placebo, but it's decrease in the tofacitinib group compared to the placebo group, suggesting that this could be pathway that targets vascular dysfunction in lupus patients. Now, we all know about the black boxes and the risk for thrombosis associated with JAK inhibitors, so this needs to be taken carefully and more studies are needed, but this suggested that this is a pathway that may have multiple roles in modulated disrupted innate immunity or pathogenic innate immunity in lupus, and also with consequences for vascular damage. So I will end here, just want to highlight a couple of points I tried to make during my talk. One is that sex differences in neutrophil biology may play a role in the differential risk for autoimmunity, and more work is needed to further understand that. That NETs are a source of modified autoantigens, and this may promote pathogenic autoimmunity in individuals at risk. That one of the mechanisms by which the NETs may be particularly pathogenic is through the enhanced oxidation of nucleic acids that may drive type I interferon responses, vascular damage, etc. And that better understanding neutrophil heterogeneity may allow us to target specific neutrophil subsets and therefore contribute to the mitigation of some of the end organ damage and immune dysregulation characteristic of these diseases. And finally, I will end here by acknowledging a lot of people, several of them were mentioned during my talks, both current and former members of my lab, both at the NIH, at the University of Michigan, my great collaborators, both at NIAMS and another NIH institutes, great collaborations with many people, including Keith and Christian at the University of Washington, as well as in other institutions, and my funding by NIAMS. So thank you very much for your attention, and hopefully I can answer some questions. Thank you.
Thank you so much, Miranda. That was a fantastic presentation. I think such a beautiful overview and some very new, interesting findings as well. Then we have a few questions. If you don't mind, I'm just going to start off with one or two of my own. You discussed the heterogeneity of neutrophils in general, in these 4 different clusters at least. What is known about the heterogeneity of LDGs? And do you think that LDGs kind of are connected with, let's say, the interferon neutrophil subtype or anything like that?
Yeah, that's a great question. Thank you, Christian. So we've been doing some single cell RNA-seq of the LDGs. It gets more and more difficult as you have less cells to study. But I think overall, some of these data we have published, LDGs are not the same. So what I think is that at least in lupus, the majority are mature neutrophils, but there is a small subset within the LDGs of what would be like bona fide immature. I mean, initially when LDGs were described, people said these are immature cells. In our experience, that's not the case. The majority are actually pro-inflammatory mature, but perhaps more in the intermediate stage of neutrophil maturation compared to, at least from what has been described from single cell studies in the bone marrow. Compared to the very, very mature cells you may see, infections or things like that. Now, the other question is whether if you look at other diseases, are the LDGs the same, right? And people describe LDGs, but then don't do a lot of functional studies. So it's hard to tell whether all these LDGs are the same. But I do think there is some heterogeneity, and how that relates to the response to type I interferons is a good question, because I think the more mature the machinery, the more they will respond. So that remains to be further studied as well. Yeah,
that was kind of one of my follow-up, I suppose, depending on, we're thinking about interferon, whether that was also present in LDGs in other conditions like myopathies or RA and all of that. And if that could be the general association or link to vascular disease in these populations.
So we're currently working on that. We're doing some comparative studies of LDGs across diseases, and we're trying to better understand that. So certainly, for example, in psoriasis, where we see LDGs, we don't see the same. And so I don't think they're all, there's something else probably that we're missing there. But
very interesting. Yeah. A few questions, if you still have a few minutes.
Yes.
There were a question here from Vivian. Do you have data on the difference on neutrophils in children with lupus, males versus females?
Yeah, we are doing those studies right now because we are very interested in pursuing further this sex difference story, but we don't have the results yet. So yeah, we want to look, because in kids without lupus, we don't see differences, but what happens in children? Yeah. That's a great question with SLE. Yeah.
Excellent. And then from the same, from Vivian again, do you think that the role of mast cells and their role on signaling to neutrophils is underestimated? What is known about that kind of interaction?
Yeah, I mean, as you know, not only neutrophils make extracellular traps, right? So in theory, any cell that has granules may have the ability to do this. We know basophils do it. Some groups have seen it in macrophages. It's been described in mast cells. And I've even seen some papers on T cells. I mean, it's a pretty ancestral way of defense, right? I mean, even plants can extrude nucleic acids as a defense mechanism, and it's been described, you know, in zebrafish and certainly in the various mammals. So what is the eosinophils can do it too, sorry. So what is the role of these different traps, and why would the traps made by neutrophils have certain roles versus what eosinophils, for example, in certain types of vasculitis, etc., may do? We had some story with mast cells and traps and psoriasis a few years ago, that we were seeing mast cells also making these traps in the skin. We didn't follow up much after that. I did that in collaboration with a dermatologist when I was at Michigan. But it's a great question. I mean, what are the mediators in those specific traps that may drive certain types of phenotype versus what happens with other cells?
Right, and also the concern, I suppose, with measuring, let's say, MPO DNA complexes in circulation and saying that this is from nets, whereas it could be from potentially from other sources.
Absolutely, and that's always been, you know, as you know, I mean, how specific can we be? And with neutrophil elastase maybe a better one, but it's always an estimate, right? Yes.
The final question before we need to end, and it continues to relate to sex differences, and the question goes: in women, do neutrophils change functionally across the menstrual cycle? And if so, what are the differences?
Yeah, that's a great question. So what I can tell you is we looked in pregnant women. And that was pretty interesting because as women became pregnant, this more mature phenotype actually went away. And during pregnancy, it mimicked more the male phenotype. And then after the postpartum, it went back to the female neutrophil phenotypes. Again, supporting this is hormonally driven. Why that happens, we don't know. I mean, could it be some sort of immune tolerance mechanism that, you know, the neutrophils become less active during pregnancy? You know, but it was pretty striking that at least at the cell, I mean, we don't know about nets, but we did look at neutrophil activation, and it did look more like male neutrophils during that period.
Very cool.
Yeah, so, but we need to do more studies about that, yeah.
Wonderful. With that, I think we have to end, Mariana, but so very grateful for your presentation and talk and discussion at the end. And yeah, looking forward to reading some of your new papers coming out as well.
Thank you very much. Thank you for the invitation. Thank you. Okay.